Hi, good morning, and welcome back to my channel, Biochemistry Quick Review and Guide. And today in this session, we are going to talk about ketone body metabolism. So, our specific learning objectives are you should be able to identify the ketone bodies both by names uh, and structure as well. Then you should be able to describe the synthetic pathway of ketone bodies in liver and mitochondria. Then, utilization of ketone bodies in extrahepatic tissues. Then you should be able to describe the conditions in which ketone body synthesis and use are favored. Then you should be able to describe the three stages in the metabolism of fatty acids where ketogenesis is regulated. And overproduction of ketone bodies leads to ketosis and acidosis. Ketone bodies. We have three ketone bodies: acetoacetate, uh, acetoacetate, acetone, and beta-hydroxybutyrate. These are the three ketone bodies. Among these, acetoacetate is called the primary ketone bodies because it is synthesized first. And thereafter, this acetoacetate undergoes spontaneous decarboxylation to produce acetone. And this acetone is a volatile gas, so it comes out through the lungs. And this acetoacetate can undergo dehydrogenase reaction by enzyme 3-hydroxybutyrate dehydrogenase. So it can be uh, reduced to 3-hydroxybutyrate, D3-hydroxybutyrate, and the coenzyme used is NADH. Uh, in well-fed mammals, normal blood concentration of ketone bodies does not exceed 0.2 millimole per liter. That's why in normal individuals, detection test for ketone bodies, that is Rother's test, is negative. But in certain conditions in which the ketone bodies are excessively produced, then the ketone bodies will, this Rother's test will become positive. When does ketogenesis occurs? That means in what condition or in what situation the ketogenesis will take place. Basically, ketogenesis occurs when there is high rate of fatty acid oxidation in the liver, and high rate of fatty acids will be produced from two conditions, that is in uncontrolled diabetes mellitus and starvation. Since in uncontrolled diabetes mellitus there's no insulin, so the glucose from the blood cannot enter the cells. Therefore, the cell suffers from lack of energy. And similarly, in starvation, since the person is not taking any food, there is lack of glucose, and therefore the body cells, they look for another source of energy, and the immediate source of energy next to carbohydrate is the fatty acids. So in these two conditions, there will be altered insulin glucagon ratio. So the insulin is low and glucagon is high. Under the influence of glucagon, there will be lipolysis. Increased lipolysis in adipose tissues produces increased amount of free fatty acids. This reaches the blood, and from blood it reaches the liver. In the liver, oxidation of these free fatty acids produces acetyl-CoA, and this acetyl-CoA further leads to synthesis of ketone bodies. So in starvation and uncontrolled diabetes mellitus, there will be excessive production of ketone bodies. Now, pathway of ketogenesis in the liver mitochondria. Ketogenesis mostly occurs in the liver mitochondria. So here what you can see is this this is the active free fatty acid activated to acyl-CoA. Okay, then this acyl-CoA enters mitochondria by this carnitine shuttle, and then or it will undergo beta then in the mitochondria it will undergo beta oxidation, and acetyl-CoA will be one of the intermediates of this beta oxidation of fatty acids or beta oxidation of acetyl-CoA. So this acetyl-CoA, this is a 4-carbon molecule, may condense with another molecule of acetyl-CoA here to form HMG-CoA, that is 3-hydroxy 3-methyl-glutaryl-CoA, in short we call HMG-CoA, by the enzyme HMG-CoA synthase. Or in your book it is simply written, first of all, 2 molecules of acetyl-CoA condense to form acetyl-CoA, and then it again condenses with another molecule of acetyl-CoA to form HMG-CoA, and this HMG-CoA then is acted upon by another enzyme that is called HMG-CoA lyase. That will split HMG-CoA into acetoacetate and acetyl-CoA. So this is our first ketone bodies, that is acetoacetate. This acetoacetate, as we have already seen, may undergo spontaneous decarboxylation to produce acetone, which will be exhaled out from the lungs, or it may undergo dehydrogenase reaction by this enzyme 3-hydroxybutyrate dehydrogenase to produce D3-hydroxybutyrate. This is another ketone bodies. So this way the ketone bodies are synthesized, and these ketone bodies and this acetyl-CoA so produced may either again go for a synthesis of another molecule of ketone bodies, okay, so or yes, these things will not occur here during ketone body synthesis. This and esterification of acyl-CoA to triacylglycerol and phospholipid will not occur when the process of ketogenesis is taking place, because during the process of ketogenesis, it is under the influence of your hormone glucagon. So this esterification and production of triacylglycerol, phospholipid, esterification with triacylglycerol and phospholipid will occur in the fat state when there is excessive carbohydrate in the diet, and that is being converted to acetyl-CoA, and then only then, under the influence of insulin, this esterification and deposition in the adipose tissues will take place. Otherwise, in the fasting state or during starvation and diabetes mellitus, this pathway will not be active because there is no insulin. So you can just simply so this is the synthetic pathway. Next, this is same. Next, pathways of transport of ketone bodies from liver and its utilization in extra hepatic tissues. So after the ketone bodies are synthesized in the liver, this is acetoacetate. This acetoacetate can directly enter to the circulation and reach the muscles, or in the form of 3-hydroxybutyrate, it may enter the blood vessels and reach the skeletal muscles. And in the skeletal muscle, beta-hydroxybutyrate may be converted to acetoacetate. And this acetoacetate is converted to acetyl-CoA. Acetoacetate combines with succinyl-CoA, which is coming from T-phase cycle, and then it is converted to acetyl-CoA and succinate. Enzyme here is thiophorase or CoA transferase. So this thiophorase enzyme is very important, and because the presence of this thiophorase enzyme, skeletal muscle is able to utilize the ketone bodies. And this thiophorase is absent in the liver, and therefore ketone bodies are synthesized in the liver, but they cannot be utilized by the hepatic cells, so the liver cells. So this is a short question you'll ask: why ketone bodies cannot be utilized by hepatic tissues? So this is your answer. Hepatic cells do not contain enzyme thiophorase, and therefore hepatic cells cannot utilize ketone bodies. Whereas it is present in the muscles, in the extrahepatic tissues, or in the muscles, and therefore muscles can utilize the ketone bodies for energy production. So now you get acetyl-CoA, which will be cleaved into two molecules of acetyl-CoA, and this acetyl-CoA can enter the TCA cycle and produce ATP through the TCA cycle. So this is how it extrahepatic tissue utilizes, or the skeletal muscle utilizes, the ketone bodies. Now, net flow of ketone bodies to extrahepatic tissues. Let us just follow the path how the ketone bodies will reach the extrahepatic tissues. So once the ketone bodies are synthesized in the liver, so in let me start from here, blood. So during starvation or in diabetic patients, there will be lots of free fatty acids which is coming from the lipolysis of the adipose tissues. So plenty of free fatty acids will be present in the blood, and this will reach the liver. In the liver, acetyl-CoA will be activated to acyl-CoA, and this acyl-CoA will undergo beta oxidation to produce acetyl-CoA. And this acetyl-CoA will go for the synthesis of ketone bodies, and once synthesized, these ketone bodies will again come to the blood, and from blood it will reach the extrahepatic tissues, and in the extrahepatic tissues ketone bodies will be reconverted back to this acetyl-CoA, and then it is utilized in the TCA cycle to produce energy. Now what are these dotted lines? Let us see what are these dotted lines. These are the other possible pathways of the free fatty acids. Free fatty acids in the extrahepatic tissues can be produced to acyl-CoAs and form acetyl-CoA. Acetyl-CoA can also come from glucose, but this will be during fed state. Okay. And this side, the acetyl-CoA can come from glucose, but this will be during fed state. So in our case, since we are talking about synthesis of ketone bodies, this is mostly taking place in the fasting or starvation or in diabetes mellitus. So we are not considering this. But these are the pathways when it will be active during fed state. So glucose to acetyl-CoA, and during fed state, acetyl-CoA may go to the TCA cycle, and it is completely oxidized in the TCA cycle, because that time there will be presence of oxaloacetate. To metabolize this acetyl-CoA, you need oxaloacetate in the TCA cycle. And once ketone bodies are synthesized in the liver, it reaches the blood, and some of the ketone bodies like acetone will be exhaled out from the lungs, and some ketone bodies may be excreted in the urine to cause ketonuria. Okay, and the excess and the rest will reach the skeletal muscles, and they will be catalyzed in the TCA cycle. Now, regulation. Factors regulating mobilization of free fatty acids from adipose tissues are important in controlling ketogenesis. So, factors regulating mobilization of free fatty acids from adipose tissues. From adipose tissues, how the free fatty acids are mobilized? First is by lipolysis. The adipose tissue triacylglycerol undergoes lipolysis under the influence of glucagon and form free fatty acids. So this is one factor. If you can control lipolysis, the ketone body synthesis can be controlled. So lipolysis is one. After the free fatty acids are formed, it reaches the blood, and from blood it comes to the liver. And free fatty acids, after beta oxidation, after activation, it forms acyl-CoA, and this acyl-CoA undergoes beta oxidation to produce acetyl-CoA. So, and further this acetyl-CoA will go in the process of ketogenesis to form ketone bodies. So from this point, this free fatty acid or this acyl-CoA has three phase. One is either it can be esterified to form acylglycerols, or it may directly form acetyl-CoA, then ketone bodies, or this acetyl-CoA may have entered the citric acid cycle to produce energy. But in during starvation or in uncontrolled diabetes mellitus, there is lack of insulin, so this esterification of acetyl-CoA to acylglycerols will not occur. Moreover, there is lack of oxaloacetate in the TCA cycle, therefore acetyl-CoA will not be oxidized through the TCA cycle to the carbon dioxide and water. Therefore, this acetyl-CoA will be going for the synthesis of ketone bodies. So these are the 3 points in the process of lipolysis, in the process of mobilization of fat from adipose tissues, which can be controlled to decrease production of ketone bodies. And that means you can either divert them. If you can divert this acyl-CoA to the esterification to form glycerols and triacylglycerols, then ketone body synthesis will be decreased. Or if you can divert acetyl-CoA into the TCA cycle, then the synthesis of ketone bodies will be decreased. But naturally it does not happen because these pathways will be under the influence of insulin, and during starvation or in diabetes mellitus there is lack of insulin, so this pathway will not occur. So let us see in this slide how the regulation of long chain fatty acid oxidation in the liver leads to regulation of ketone body synthesis. Let us try to understand this flow chart under 2 situations. One is fat state, one in fat state and one in fasting state. Let us see in fasting state. In the fasting or starvation or in diabetes mellitus case, there will be excessive production of free fatty acids from the lipolysis of adipose tissues, and this excessive free fatty acids will reach the hepatic cells in the liver, and in the liver they will be activated to acyl-CoA, and this acyl-CoA will go into the mitochondrial matrix and produce undergo beta oxidation and then produce ketone bodies. Okay. Now, during this fatty acid oxidation, acyl-CoA is formed, and this acyl-CoA can inhibit one of the enzymes of lipogenesis, that is acyl-CoA carboxylase. It means that during starvation, fasting, or diabetes mellitus, there will be no lipogenesis taking place. Okay, there will be no lipogenesis taking place. Now, let us see in the fat state. During fat state, there will be lots of glucose in the blood, as well as there will be free fatty acids also some. So this glucose will be ultimately converted through lipogenesis to the free fatty acids, that is palmitate or palmitoyl-CoA. The fatty acids will be formed. And during fat state, there is presence of insulin is there, and this insulin will stimulate this enzyme acetyl-CoA carboxylase, which is important enzyme of lipogenesis. Okay, so there will be increased synthesis of the fatty acid during your fat state. And during fat state, this malonyl-CoA, one of the intermediates of lipogenesis, it also inhibits enzyme carnitine palmitoyl transferase 1. So it means that during lipogenesis, there will be no beta oxidation of fatty acids. That means these two pathways cannot occur simultaneously. In the fat state, lipogenesis will take place and leading to the formation of fatty acids. And in the fasting state or in the starvation or in diabetes mellitus, there will be beta oxidation of fatty acids, but there will be no lipogenesis. So these two pathways will take place in different conditions. One is in fat and one is in fasting state. Okay. Now, during fat state, there will be some amount of free fatty acids in the blood, and these free fatty acids will be esterified with acylglycerols to form acylglycerols, which will be incorporated into VLDL, and they will be transported in the blood vessels, in the blood circulation to the different tissues in which they will be utilized. So that is the basic thing. Okay, that's all. So what is ketosis? Ketosis is a condition in which there is excessive amount of ketone bodies in the blood and excretion of ketone bodies through the urine. That is called ketosis. In normal individuals, the blood level of ketone bodies is less than 1 milligram per deciliter. And this only traces amount of ketone bodies are excreted in the urine, which are not detectable by Rothera's test. Rothera's test is the test which detects presence of ketone bodies in the urine. But when the rate of synthesis of ketone bodies exceeds rate of utilization, there is accumulation of ketone bodies in the blood, that is called ketonemia, and therefore there will be excretion of ketone bodies in the urine, that is called ketonuria. And also there will be excretion of some acetones through the breath. So the breath smell will be that of acetone. Now, what are the causes of ketosis? Ketosis is caused by these two conditions. Ketosis is caused by uncontrolled diabetes mellitus and prolonged starvation. Now let us see how in diabetes mellitus ketosis is produced. So in diabetes mellitus there is insulin, no insulin, no insulin, no insulin. Therefore, the glucose cannot enter inside the cells, and therefore there will be no oxidation of glucose to produce ATP, and the cell lacks ATP. So the cell looks for another source of energy, and fatty acids are the another immediate source of energy for the cell. So in uncontrolled diabetes mellitus, since there is no insulin, there will be increased amount of glucagon hormone, and under the influence of glucagon, there will be lipolysis. It will cause lipolysis of triacylglycerols in adipose tissues and produce free fatty acids, and these free fatty acids undergo beta oxidation in the liver to produce acetyl-CoA by beta oxidation. And this acetyl-CoA will ultimately produce ketone bodies, and these ketone bodies will be exported out from the liver to the extrahepatic tissues and utilized through TCA cycle to produce ATP. That is how the cells are getting energy from the lipids during uncontrolled diabetes mellitus. Similar condition is for prolonged starvation. In prolonged starvation, there will be no glucose in the blood. There is no glucose in the blood. No glucose. No glucose. Therefore, there will be lipolysis. Therefore, there will be lipolysis because there will be increase in glucagon, and that will cause lipolysis, and the situation and the same thing will follow free fatty acids will form, then acetyl CoA will form, then ketone bodies, and ketone bodies exported to the extrahepatic tissues and they are utilized to produce energy through TCA cycle. So that is how during starvation the cells are getting energy from the free fatty acids. Thank you. Thank you all.